Welcome back to the Business of Biotech. I'm Matt Pillar, Chief Editor at Bioprocess Online, and your guide for today's conversation with Cartesian Therapeutics co-founder and CEO, Dr. Murat Kalayoglu. On today's episode, we're going to focus on GMP expectations for phase 1 first-in-human trials. And Dr. Kalayoglu is without question a fitting guest for the topic. Prior to the launch of Cartesian Therapeutics, he co-founded a company called Topokine, which he led through late-stage clinicals before selling to Allergan. Now at Cartesian, he's managing multiple clinical trials. Dr. Kalayoglu earned his MD and PhDs in immunology from the University of Wisconsin-Madison, followed by a Harvard residency and an MIT Sloan MBA. Dr. Kalayoglu, I'm thrilled to have you on the show. Welcome.
It's great to be here, Matt. Thank you.
Yeah, well, I'm thrilled you could join us, and I hope I'm getting the name, the pronunciation right. As I said, I'm sure to butcher it at some point through our conversation. So
you nailed it, Matt. You absolutely nailed it.
Excellent. So I want to start by learning a little bit more about you. Excuse me. On paper, it looks like your career trajectory sort of took a pretty distinct turn from academia into industry after your residency and fellowship at Harvard, albeit with a bit of a twist in that it looks like you dabbled a little bit in behavioral health software at Health Honors. I didn't mention that in the intro because it's maybe not as relevant to a phase 1 clinical trial conversation. But tell me about your story. So we see a lot of folks come out of academia after a time and kind of take a meandering path into, entrepreneurship. You sort of sprinted towards entrepreneurship right out of academia. Was that intentional? Was that circumstantial? Give us the backstory there.
Well, I mean, growing up, my father was a doctor, he's an academic physician. And so I always felt that what he was doing was worthy because he was very happy with what he did. And, as a surgeon, he'd come home in the middle of the night and just be happy, right? And so I figured that was probably a path that I should at least consider. So I recognized sometime in college, as I was doing research in immunology and microbiology, that I wanted to do something along with research. And so I thought then this integrated MD PhD program was probably the best way to sort of be able to do both, see patients as well as do research. So started going down that path. And somewhere along the way, realized that the actual day-to-day practice of medicine was probably not a good fit for me. And there's a lot to say about that. We'll probably just skip right over that. But I really enjoyed the research part of it. So, the decision for me was after medical residency, whether to go and become a researcher, an academic researcher, or do something different. Around that time, during residency, 2 important things happened. Number one, my residency program director, Tony Adamus, founded a company called iTech, an ophthalmology drug for macular degeneration back in the early 2000s. And that really sort of influenced me because I realized, boy, you could do that. I didn't realize that you could actually sort of leave academic medicine and sort of do something different. And the second thing that happened was I met my longtime friend, colleague for the last 18 years now, Mike Singer, who was in the same residency program with me in ophthalmology at Harvard. And he and I sort of thought in similar ways that we wanted to do something to contribute, but we perhaps recognized it wasn't going to be a career in academic medicine. So we joined forces, and over the next, many years, ended up, starting a couple of different ventures. The first one was in behavioral economics, as you mentioned, healthcare IT. This is a way we developed a platform, the technology to motivate people to remember to take their medications online. And through using some strategies through Skinnerian conditioning and intermittent reinforcement in behavioral economics, that platform expanded to include other healthy behaviors. And so we developed that over several years from 2006 to 2009. And in 2009, went to an original observation that we and other colleagues at Harvard had made around the use of glaucoma medications for reducing fat. And so we and others made the observation that these eye drops that we would instill in our patients with glaucoma had the untoward side effect of reducing fat around the eye. And so we took the active molecule of that, which is a prostaglandin F2 alpha, and made it into an ointment form and applied it directly to the skin to reduce something called steatoblepharon, which is fat under the eyelids, as well as other parts of the body. We were going after the fat in the neck and the buffalo hump in some patients with HIV lipodystrophy and other parts. And we took that from concept all the way to phase 2b3 registration studies before handing it off, as you said, to Allergan. And then a week later, started Cartesian. And Cartesian was really born out of almost a necessity we felt. My brother was diagnosed with Hodgkin lymphoma. Right around that time that Topokine was, nearing the end and its sale. And I recognized that a career in, medical aesthetics was probably just not a good fit for me at that point. So, and Mike had also a family member who was diagnosed with myeloma. I had another family member diagnosed with myeloma. And so we went hunting for the best technology out there. And licensed some core technology from the NIH that allowed us to sort of get going to try to develop a potent yet safer therapy to be able to be used in patients who were newly diagnosed because we felt that that's the best time to intervene. It's always easier to treat a disease earlier in the disease process than it is later in the disease process.
Yeah.
So, so this happened to be a cell therapy, it happened to be a CAR T therapy. And we had no idea how to go about doing it, but we recognized the potential of the technology. And so that's how Cartesian was born in middle of 2016.
Yeah, it's that's an interesting pivot, from, as I said, it looks like you raced into entrepreneurship, but I guess the path was, kind of meandering in fact, at the end of the day, because you went from, IT, so to speak, a software application to, aesthetics to, biologic therapy. That's a pretty big leap.
We've tended to be opportunistic along the way, and really sort of flexible as to how that meandering river tends to sort of push us in one direction or another until Cartesian. And I think there is something to be said about balancing a degree of opportunism with a real conviction and an innate desire that is driven by, personal motivation. Yeah, that sort of helped us really focus and build, a platform that I think has unparalleled opportunity, and not just in oncology, but now beyond oncology and autoimmune disease, respiratory, cardiovascular, et cetera.
Mm-hmm. Yeah. And we'll get into that. I just want to reflect real quick on something that you said around, you leave a lot to the imagination when you say that you chose not to go down the sort of clinical physician route for some, reasons you don't necessarily care to disclose. I'm just going to go ahead and assume that you're afraid of needles or blood or both. At any rate, so let's talk about those the programs. You've got 6 autoimmune, oncology, and respiratory candidates. Is that correct in your pipeline? Half of those at phase 1 or beyond.
Yeah. We have 3 clinical programs in phase 1-2 studies, and then 3 preclinical programs. At least 1, if not 2 of those, is likely to matriculate into the clinic sometime in 2021. Mm-hmm. And then a real effort to continue developing new candidates as we expand the platform. The one thing I'll say is that the platform, we coined the term RNA armory to this platform. We focus only on mRNA-engineered cell therapies. Mm-hmm. And the vision behind the platform, the RNA armory, is to mRNA engineer any cell to deliver to any tissue, any combination of therapies. So, imagine it as a highly versatile cell therapy platform that is geared towards delivering not just one therapeutic, but multiple therapeutics directly to the site of disease, where we're using the cell as not just the factory of production, but also the vehicle for delivery of this combination therapy. And so because it's a combination therapy platform, the number of iterations and combinations you can come up with is really limited by your imagination.
Yeah.
And so it's a lot of fun, but it's also a bit overwhelming at times to sort of recognize all the different ways that science today teaches us to be able to target a disease that is, complicated. It's not just sort of, diseases often don't respond to treatments with a single drug. And so the ability to use cells to be able to deliver combinations of therapies opens up a whole world of different possibilities and keeps us busy.
Yeah. And from a business standpoint, it sounds like, that's a sustainable platform. There's a lot of different directions that you can take, and many shots on goal, as they say, right?
Yep.
So I want to ask you some questions around. So keep in mind, I'm going to ask you these questions, and I want you to respond to them in the context of, you've been down this path, and our audience is primarily, there are a lot of first-time leaders of new and emerging biopharma companies, biologic therapy companies. So I'd like you to kind of address these questions in that context, as though you're having a conversation with peers who maybe haven't seen some of the stuff that you've seen as it relates to going into a phase 1, first in human, clinical trial. So let's talk about that. Sort of that GMP expectations around those trials. First of all, and this is a big kind of sweeping question that I'm going to invite you to take any direction you want to. But what's your take on the FDA's guidance documents for phase 1 clinical trials? Sort of give us a brief overview of what they are and how they do, may or may not help prepare you for what you're about to see.
Sure. Yeah. I don't necessarily consider myself a regulatory expert, but certainly, my take overall on guidance provided by regulatory agencies in general and FDA in particular is that it's appropriately broad to give guidance as opposed to specific instructions. I think that from a regulatory perspective, there is recognition that there's a lot of different ways to end up at the same goal. And so the guidance documents very rarely, indicate that, something has to be done according to this set plan. That's the whole purpose, is to give guidance as opposed to instruction. Mm-hmm. And I think the companies that do well take the guidance to heart, but tailor it to meet the demands of their team and organization with respect to whatever indication they're going after and the kind of products that they're developing. The guidance around small molecules and biologics have, been around for many, many years, and it's developed over time to have more structure. Cell therapy just isn't there yet. We're really at the very early innings of this very long game, and I think there's a lot of talk to sort of improve upon processes, both from a safety perspective, production perspective, in order to ensure that we can, implement the best products that are consistent and reliable. The specifications, change over time as people get to know their products, mm-hmm. And the processes, continue to develop. And there's this overarching effort to try to close as many steps as possible during your manufacturing process. But sometimes it's just not possible for cell therapy, right? Depending on the kind of cell therapy you're doing, it's sometimes not possible. So I guess what I'm suggesting is that the guidance is a good start. But ultimately, you've really got to use your own judgment in terms of what product you're trying to develop for what purpose. And then take the guidance and everything else about keeping a product safe and sterile, and apply it in the context of exactly what you're trying to do as opposed to what someone else is trying to do.
Mm-hmm. Yeah. Yeah, and so that the fact that this is an emerging space for sure plays into your response to these questions. I mean, I talk with a lot of biotech leaders in the cell and gene space who talk about, when we work with the FDA, it's truly working with the FDA. That guidance is, in many cases, it's guidance because the FDA hasn't seen some of this stuff itself, right? So, do you find that to be the case? As you move forward, it's more of a less a transactional relationship and more of a partnership.
Yeah, no, absolutely. For the most part, the folks that deal with the cell and gene therapy space over there are overwhelmed with the number of such conversations that are ongoing, and we've dealt with across the years with many different divisions, and we've certainly found that the cell and gene therapy division, the OTAT folks, are clearly the most engaged for any of the other divisions we've ever worked with, and it's really a pleasure. But I think it also speaks to the fact that you do need this type of conversation, and you do need this type of sort of back and forth engagement at the early stages of entire sort of industry that is spawning over the last decade.
Yeah. So, and again, this question may be sort of forming as we forming up, and the answer to this question, that is, maybe forming up in real time. But what are we learning, I guess, about the key elements of GMP for phase 1 clinical supply in the cell therapy space?
Yeah. So first of all, we're learning that it's not easy. It's not easy to institute consistency between lots when the cells are derived from different sources and different tissues and different people, right? There is an enormous amount of variability that we try to control. We sometimes sort of delude ourselves in thinking that we've sort of achieved the kind of sort of delta that is acceptable. But sometimes one of these lots just throws something at you've just never seen, and you just have to sort of deal with that, right? So there is a question as to whether you could ever make cell therapy manufacturing, as consistent as, say, manufacturing monoclonal antibody, right? And I think in 2021, the answer is the jury's out. I mean, I think maybe if you asked it 5 years ago, it would have been a clear no. But there's clearly work. I mean, this is one of the reasons that a lot of people, including us, focus on allergenic manufacturing to sort of create single lots out of. But at the end of the day, if that's a single lot, that you're making another sort of lot from another sort of vial from your master cell bank, the degree of control that you can institute is still going to be limited, and it's going to be even less so relative to if you're going to grab some new tissue and to create a new master cell bank in ways that otherwise wouldn't be such a problem when sort of traditional biologics manufacturing, small molecule manufacturing. Because again, you're grabbing new tissue with different and really uncharacterized properties in terms of cell expansion, in terms of cell function, and these kinds of things. So the critical parameters they have to do with the inherent functionality and expansion potential of the cell. Everything else you can try to control, but there are some inherent properties that we just don't know about. I mean, we're in essence manufacturing tens of thousands of proteins within a given cell at any given time to sort of predict their behavior and how they interact with one another when we take them into these completely artificial environments is asking a lot relative to manufacturing a single purified protein.
The Business of Biotech is brought to you in partnership with Cytiva. Together, we're committed to helping the leaders of new and emerging biopharma companies navigate the financial, organizational, human resources, and regulatory waters you'll encounter on your way from discovery to the clinic and beyond.
Check out a host of useful resources for biotech leaders at Cytiva's Emerging Biotech Accelerator at cytivalifesciences.com/emerging biotech. That's CYTIVA lifesciences.com/emerging biotech.
How much tolerance is sort of built in to the equation here? You talk about the variability and the difficulty from lot to lot achieving consistency. How do we accommodate for that variability? Is there tolerance built into sort of the GMP equation?
Well, there is, but I mean, I think we, as well as others, do our best to minimize, that variability in ways that we can, right? And there's a lot that we can do, right? I mean, this is why, very sort of specific SOPs are absolutely critical. Minor things in the batch record make a huge difference. Mm-hmm. And unless you actually have that experience as a manufacturing organization in terms of both volume as well as a team that you built in order to sort of think through the implications of each step, you're just not going to learn from each of these steps the way that you otherwise would. You'd be missing a huge opportunity to reduce the variability and create the very best product possible. This is one of the reasons why when we built our team, every single one of our operators in GMP manufacturing are either MDs, PhDs, or MD PhDs, mm-hmm. right? So there we look for people, even at some of the most basic steps along the path, that have been formally trained to critically think, right? And then we bring them in, we teach them the process. We don't need them to have 20 years of operating experience and sort of making GMP product.
Yeah.
We want them to be critical thinkers because we don't want to waste an opportunity for manufacturing a given lot, because that's an entire patient's lot there, right? I mean, if it's an autologous product, that product is going to a patient and could potentially be, remarkably effective, potentially curative for that patient. We don't want to miss that opportunity. We don't want to miss learning as much as possible from that opportunity and also make that lot as perfect for that patient as possible. So we bring people that can critically think and learn from each and every step along the way, because sometimes these minor changes in the batch record that sometimes are, things that are not possible to capture entirely in an early stage manufacturing effort in a batch record. Mm-hmm. are things that can have dramatic implications in terms of the quality and the consistency and the variability of the product.
Sure. Yeah. Well, that's a good segue into the question I had for you around the components for the CMC information required for INDs in cell therapy. What are the key components around chemistry, manufacturing, controls that you would share with your peer emerging biopharma leaders?
Yeah, well, I mean, I don't think they're any different at a high level relative to manufacturing anything else. I mean, so, the CMC section of an IND is, 32S, 32P, and 32A if you've got your own sort of facility. So, often these are sort of continuous manufacturing processes that sort of flow from an S to a P, as opposed to the traditional sort of small molecular way of doing this, where you sort of create the drug substance and then, describe that in detail, and then sort of have a completely separate section in product. Often cell manufacturing has as a sort of a more sort of a continuous process, a more fluid line between the two. So the kind of critical sort of process parameters are similar, I think, between small molecular biologic versus cell therapy in terms of the ways in which you try to sort of control that process, sample it along the way to ensure that you're, learning as much as possible every step of the way in terms of how your cells are behaving. You check for sterility along the way, you check for, you check for potency, you check for function, and then ultimately, you do your full release testing at the very end and compare that data relative to what you've generated in order to come up with, appropriate correlates to optimize on your product. So that process, again, takes a lot of thinking, a lot of planning ahead, and a lot of processes to ensure that you're extracting as much information as possible during the development.
Yeah. Just as an aside, out of curiosity, is Cartesian manufacturing clinical supply in-house, or is that outsourced?
Everything we do is in-house. So we're a fully integrated company in that we do not just GMP manufacturing in-house. We have our own quality management systems in-house. We release product here internally. We have our own clinical quality control laboratories here. We do preclinical development, discovery all here, and then we also do clinical operations here. So everybody's under the same roof. Mm-hmm. And that organization allows a lot of cross-divisional communication between folks that otherwise wouldn't necessarily be able to talk so quickly and fluidly between departments, between each other. Yeah. And we bring folks to the team that have a curiosity and interest to be able to work cross-divisionally instead of just sort of, a single role. And we've got, somebody on an MD on a clinical operations side can jump in and actually do GMP manufacturing. That sort of, a director of research could go and contribute to the quality, could contribute to, manufacturing or clinical operations. So that kind of integration that we've found is really important, and from a manufacturing perspective, is absolutely critical to have your own facility in cell therapy. We started off this path with a CRO that was very motivated and actually did a fine job, but we recognized that it just was not going to allow us to be able to learn as much as possible for each and every engineering and production run until we brought everything in-house. So we invested back in 2018 into a state-of-the-art GMP manufacturing facility, and then built the entire company around it.
Yeah, not the first time I've heard that story. It seems there's a trend there. Reeling it back in after trying a CRO, specifically in the cell therapy space. Let's talk about raw materials management. So, this is another unique one to cell therapies, given that, for every cell therapy, maybe not every cell therapy company that I've talked to, but there are many variables around, harvest in cell therapy. In your case, is there a raw materials management expectation in the context of GMP production?
Sure. Yeah, no, absolutely. I think, those steps need to be controlled every step of the way. I mean, we obviously try to outsource, to insource from manufacturers that regularly, generate GMP grade material. There are obviously different degrees of risk, with the highest risk that, for the raw materials that are touching the cells directly, and sort of working your way downwards to, certain types of equipment or processes that are not directly in contact. And so you sort of have to sort of use your own judgment. And then you also want to do things phase appropriately in order to ensure that you're not, unduly burdening your manufacturing process, because it does matter that the length of time that a given sort of step takes is not innocuous, right? I mean, the longer the steps are, the worse the product ends up, coming out. And that's been sort of proven over and over again across many different manufacturing steps. So you want to do things in a phase appropriate manner. You want to make sure that you've got a very robust quality management system that's able to sort of, integrate, improvements upon, raw materials as they come up, and be able to sort of, establish, a lot of the critical process attributes along the way. I think one of the key issues in cell therapy is just the relative scarcity of reagents and materials relative to the more established sort of small molecule biologics world. Yeah. So, that's something I think it keeps all of us up at night, especially during sort of the pandemic, where a lot of that sort of the sourcing issue for some of these materials has been amplified. So we try to plan as much ahead as possible. I mean, we don't have unlimited space where we can sort of store things, but we try to plan ahead and keep up with our expectations and try to bring, material in-house so that we don't get caught off guard when we've got, a busy period during our manufacturing.
Yeah, so it sounds like there's a potential supply risk there. While we're on risk, is there any other risk management advice that you'd share in the context of manufacturing early clinical stage material? Any other, I guess, pointed areas of potential risk?
Yeah, I mean, I think when you're first getting going with a process, you've really got to get your process down with as many engineering runs as possible, and do not assume that the kinds of data you're generating at small scale are going to translate into large scale, right? Mm-hmm. I see people getting caught off guard there because cells just don't necessarily behave the same way that you would expect them. And a lot of vendors, purport to have these highly scalable technologies, whether it's in just wells or transfection reagents or materials and equipment. It's not always necessarily the case that the process that works in their hands at a small scale would translate into working effectively at a large scale. So you sort of have to plan for the unexpected, really expect the unexpected all along the way, and do much of your optimization as much as you can at as large of a scale as you can in order to vet out all the unexpected issues that you're going to encounter.
Yeah. Okay. So we are running a little short on time, but in the time we have remaining, Dr. Kalayoglu, I want to make sure that we have an opportunity to talk about some of your products. And I guess I want to ask you a question about one specific product before I kind of give you an opportunity to share whatever news you'd like to out of your pipeline. And that's, that's the candidate for ARDS. And my question is, so in Q4 2020, you guys announced a clinical trial of RNA-engineered cell therapy for acute respiratory distress syndrome and COVID-19. Now, obviously, this is timely. Here, what, 5 years ago, 6 years ago, this company's in its formative years. You're focused on oncology. You're focused on some other, indications. And all of a sudden, here comes the pandemic. Here comes COVID. You find that you're well suited, perhaps, to develop a candidate to solve some of these urgent needs. Did it kind of knock you off a little bit, though? I mean, did that, like it did so many in the biopharma space, like kind of take your eye off the target and force a pivot? Tell us a little bit about that. I mean,
it was, you sort of have to do something when you're in the middle of, a worldwide pandemic. And the scientists here internally said, look, we've got this platform and we've got this disease. We know something about this disease. We can probably use our technology to be able to do something about it and probably able to get it into the clinic as, pretty quickly. So the entire process of from concept to getting, having an IND allowance took 4 months, which I think speaks to the potential of a platform like this to be able to, as you suggested, just keep generating, products for clinical testing. But I think you sort of have to respond in some way. Yeah. And we felt compelled to be able to do our part, to be able to do something, not just about the pandemic, but also, about other causes of respiratory distress, and as well as, this particular, target that we're going after, neutrophil extracellular traps, NETs, are implicated in a whole slew of diseases beyond respiratory disease, including cardiovascular disease, autoimmune disease, and et cetera. So, we felt that the opportunity was right and that the expansion potential for us to sort of make an entry into this field and respiratory gave us, enough justification to be able to pivot to address the needs. but we still, used the platform, right? It wasn't anything sort of brand new. It was a different type of cell, but the platform is agnostic to the type of cell that we can work with.
Yeah.
It was so it was an allergenic off-the-shelf therapy, but again, the platform is agnostic as to whether it's able to work with autologous versus allergenic off-the-shelf types of therapies. So this is a therapy where we're engineering two different proteins that into a single cell, and then administering that cell. This is mesenchymal stem cell therapy directly to intravenously, where the MSCs travel to the lungs, and then right at the site of disease, where these nets are abundant and is physically occluding the alveoli and causing respiratory distress in the microvasculature and causing these microthrombi. We're able to just deliver the cargo, the therapeutic cargo, directly to the site of disease. So, we're very, hopeful and optimistic about this approach. And again, it was built on the back of a platform that we had developed that was just ready and waiting to sort of take the next step, and it gave us an opportunity to be able to do that.
Excellent. Yeah. What other clinical programs would you like to share an update on? Anything else that any particular movement, I guess, recently on any of your other candidates?
Yeah, so we have 3 programs in clinical development. These are all mRNA-engineered cell therapy. By the way, the reason we focus on mRNA is to overcome the limitations of what we consider to be in conventional engineering. Conventional meaning sort of 99% of what you hear about in cell and gene therapy is DNA-based. So people are sort of making edits at the DNA level and thereby conferring upon these cells permanent changes. And so that every time that cell encounters this target in the body and divides, it doesn't know when to stop, because the daughter cell looks identical genetically to the parent cell when the cell is dividing. And so because of that, you get all sorts of toxicities, both short-term as well as long-term. So short-term, the classic example of this is CART therapies. If cell starts dividing, it doesn't know when to stop. So you get the cytokine release syndrome, neurotoxicity. And then, of course, long-term risks are, risk of transformation, or the risk of long-term immunogenicity. And while these cell types are potent, the risk profile relegates them often to use only in advanced cancers and relapse refractory cancer. So if you want to break out of advanced cancers and into frontline cancers beyond oncology, you have to have a different approach. And our approach is mRNA-based, where mRNA is a defined half-life. I think, a year ago, a lot of people didn't know about mRNA, and we can thank our colleagues at BioNTech and Moderna and Pfizer to sort of, bringing out the safety aspect of mRNA and how powerful of a therapeutic it could be.
Right.
So we confer upon these cells a defined half-life, predictable pharmacokinetics, and with that enhanced safety, we can go not just beyond advanced cancers and frontline cancers, but also beyond oncology, in our case, in respiratory and autoimmune disease until recently. So all of our products are focusing on mRNA engineering cell therapies. Our first generation products, Descartes 8 and Descartes 11, are targeting myasthenia and autoimmune disease, and then multiple myeloma and frontline cancer. In our second generation product, we talked about Descartes 30, that's our ARDS indication. And our third generation products, where we're going to be engineering, these cells with 3 or more different protein therapeutics, targeting them specifically at the site of disease. Descartes 15, 25, Descartes 33. These are products that we hope to see in the clinic, very soon. So lots of progress on lots of different fronts. No sort of new news that I can share with you at this time, but we're making steady progress. We're hopeful and optimistic about the potential of the platform.
Awesome. Yeah. Well, we'll continue to pay attention, Dr. Kalayoglu. I appreciate the time that you've spent with us, and I especially appreciate your willingness to talk a little bit about your experiences with phase 1 clinical trials and maintaining GMP clinical supply. I think your words hopefully remove some of the fear, uncertainty, and doubt that some of our first time seeing this kind of biotech leaders are seeing. So it's very much appreciated.
You got it. Thank you. And thank you for your interest, Matt. Really appreciate you doing this.
Yeah, our pleasure. Our pleasure. So that's Dr. Murat Kalayoglu. I'm Matt Pillar, and this is the Business of Biotech. We're produced in partnership with Cytiva, a company committed to helping emerging biotechs navigate the early clinical and regulatory waters, and offering resources to do so at cytivalifesciences.com/emerging biotech. Check that out. Check us out at bioprocessonline.com, where you can sign up for my always relevant and never spammy newsletter. And in the meantime, if you like what you heard here, subscribe to the pod so you don't miss an episode. Give us 5 stars. And thanks for listening.